and action.
Okay, so this is a BSL2 room. Not everything that we do in here is BSL2, but best practice is to assume that it's BSL2 and use proper precautions. So let's just go through some of the safety stuff real quick. So biohazard waste bins are over here. We have a big bin and we have a small bin that has like a foot pedal. So this is where you would put all of your waste that you generate when you're using the BSC cabinet. And if you need extra bags, they're under the sink here. So yeah, they're down at the bottom. And if you need more, and we don't have any more there, you can get them from the facilities office upstairs. Okay. We also have a sharps container over here. So this is our sharps container. So if you're using needles, they have to go in here. And these yellow containers are for prion-like proteins. So if you're working with prions or prion-like proteins, you'd use these containers. If you don't know what they are, then probably you don't need them. Okay. So that's pretty much all the safety stuff. All right, so let's just talk through where stuff is. Okay, so behind you are lab coats that we use. We typically use disposable lab coats when we're working with BSC, and so you can find them kind of throughout the room, but mostly they're on the bottom shelf over there. I would try to dispose of them like, I don't know, like every 2 weeks. Yeah, because they get pretty gross and you can contaminate your cultures. So just want to constantly be recycling those. And this stuff here is all like backup stuff, so backup dishes, whatever you can imagine, this is all backups. We also have autoclaved water over here. So this guy, because we use it to thaw cells, we like to keep it as sterile as possible, so we fill this with autoclaved water and we add Sigma Clean, which I think is actually outside, but you'll add like a certain amount of Sigma Clean. It says the dilution on the bottle, and that just ensures the fungus isn't growing in this. So we can use the autoclaved water for that, and then we also use it for the incubator. So let's talk about the incubator. Okay. So this is our incubator. It's quite new, so it should be in good working order. There are a couple things to note. So this panel here tells you what the set points of everything are and if there are any alarms that are currently going off. So it's set to 37 degrees Celsius and 5% CO2, and it does a pretty good job of maintaining that. I imagine that it will alarm at you if these kind of shift. And then it also will show you if there's water present in the incubator. So you have to keep a certain humidity level. So we do need water in the incubator. And it'll tell you whether or not you have water. And currently, it's giving us an alarm that says, please replace the HEPA filter. So we'll do that. But you would check the bottom here for any alarms. And then the Sterirun is what is used to sterilize the incubator. So you obviously don't want to do that when you have cells because it will kill your cells. It basically cycles through very high temperatures to kill anything that might be growing that we don't want. So we'll do that every so often just to make sure that we're not growing fungus in the incubator. The important thing, and I've left a note here, is if the water levels get low or they run out, then it won't like add CO2 back into the incubator and cultures will start to die. So if that happens, it will alarm and it'll be loud. And then what you want to do is add the autoclaved water and you'll add Sigma Clean. So I'll open up the incubator. Usually we spray our gloves with 70% ethanol so that we're not introducing, bacteria, fungus. So if we open the incubator, we've got a couple things of note. On the bottom here, this is where the water goes. So if I lift this trap, there's water underneath and it's getting kind of low, so when the HEPA filter comes, I'll replace all of that. But that's where the water goes and it drains out through here. So when you run a SteriClean cycle, you'll have to drain out the water first. And then the HEPA filter is all the way in the back and it's hard to see, but it's just like a little black box back there and it has a filter piece underneath. And some of that also has to be removed when you run the SteriCycle or it'll melt. Okay, so that's what happens when you keep it open for too long. It doesn't like that, so it'll start yelling. And then you can kind of hear the CO2 pick up again when I close that. Okay, so that's the incubator. That brings us to the cabinet. So this is the BSC cabinet. It's quite big, so it's a little funky. A couple things to note here. We kind of always leave it on, so you'll never really need to touch any of these buttons, but the main things to note here are the light and the UV. So if I want to work in the cabinet and I want light, then I can click that button and the light goes on, and then I would lift the sash gentle because that's how you break the screen, which has happened. So we just avoid that. So just be gentle as you're kind of lifting and closing, and if things get stuck, then call for help and we'll try to get it back on the tracks. Okay, so basic hood information. We have pipettes here that never come out of the hood, okay? We take them out very, very rarely. Really, they should stay in the hood. This pipette gun belongs to the hood, so please don't use it for bacterial work. Everything in here needs to be mammalian cultures only. And the charger is over there, and you have to wiggle the charger a little bit. It doesn't charge all the way, so just make sure that it's charging before you leave it. Otherwise, it'll just sit there all weekend. This is our vacuum line here. So this tube has to be attached to the vacuum down here. It pulls liquid into here, and then it's attached to the vacuum line through this bottom part here. So this we bleach when it gets full enough, and we also add bleach when it's empty to make sure that nothing's growing here. And then once it's been bleached for about 30 minutes, we can open it down the sink. Vacuum line is here, so you turn it on. If the vacuum line isn't pulling, it's probably because you need to change the filter. So the filter is here. It's this like round piece, and it does get kind of gunked up, especially when we work with tissues. So we have extras in some of the drawers. I can put those out if needed. Okay, when you start working in here, these lights turn green once everything is ready to go, and then you can start using the hood as you normally would. Typically, I would spray down the hood once with ethanol at the beginning, and then when I'm done, I'll spray everything down with ethanol, throw away all the trash. You can spray this guy again afterwards, and then you would just close the sash. So that guy, and then you can turn the light off, and you can turn the UV on, and I'll turn it off for now because we're standing in front of it, but the UV will go off by itself after it's kind of done its thing, so you don't really have to worry about that. Okay, last thing for this room is the microscope. It's a very basic model, but it is extremely old, as with everything else we own, so just be really gentle with it. It used to like click off when you turned off the light, but now it doesn't do that anymore. So just like you can see the power here, as I like turn this wheel, power comes on, and then it takes a while to like turn all the way off, but try to turn it all the way off so that it's not like burning out, because it's kind of a pain to replace the light. I don't even know if they make the part anymore, so yeah. Okay, I think that's everything. Did I miss anything? Okay.
No, like I think that's, yeah. Unless you want to talk about like how to put things in and out of the, like into the hood, spray them in.
Oh yeah, yeah. So anything that you're bringing in and out of the hood needs to be sprayed in on the way in. So usually cell dishes are fine because the incubator is sterile and they're just coming straight from the incubator to the hood. Anything that's not kept in like a sterile container probably should be sprayed in. So any dishes, any Falcon tubes with media from the fridge, that all should be sprayed in. It doesn't have to be like dripping wet with ethanol, but you want to, spray it down. And it is considered best practice to use a gown when you're working. If you don't want to propagate the cells, then I guess it doesn't really matter, but if you are planning on maintaining those cultures, I would wear a gown and assume that, just use best practices with sterile technique. And because we share an incubator, it's kind of on all of us to make sure that we're not spreading fungus around. So if you see problems with contamination, just let somebody know and we can try to troubleshoot it. I've had like contaminated media before and you can't always tell until it starts growing. So yeah, just be courteous because we're all using the same sort of incubator.
Do you also spray in sample when you put it in the incubator?
I don't.
Okay.
I guess that's not fully true. I don't if it's only been here or the hood. If it's been somewhere else, then I probably will spray it in. Yeah. Is that what you mean?
And then.
Oh yeah. Okay. If you want to bleach cells, like if you're just done with them, so we keep 10% bleach here. You should kind of refill it every week because bleach doesn't last that long. You'll be able to tell because the color of the media will change. So when you bleach the cells, it should change from pink to like kind of purpley white by the end. So you can let that sit for about 20-30 minutes and then you can just dump it down the sink. And they should be dead. So yeah, if it doesn't look like the color is changing, then you probably just need to replace the bleach. We keep bleach under the sink. This is the 70% ethanol jug. We refill it whenever it runs low, and you can use this to refill the spray gun. And there's something else I was going to say, but